Hello, everyone. My name is Serge Roux. I'm the Equipment Director and Chief Designer here at NETRI. I'll be your host for this evening, and this is my co-host, Helen.
Hi, everyone.
So Helen is our electrophysiology expert, resident expert, and she'll be backing me up for any hard questions you might have for us. So let's begin. So this is number 4 of our capabilities sessions. This is like a 15-minute session with 15 minutes of me presenting what we mean by functional activity, and then maybe some questions and maybe a little bit of Q&A. All right, so let's get going. So first, who we are, for those of you who don't know us. So we are NETRI. We basically design and manufacture and sell neuro organs on chip. Okay, and we are focused on trying to improve the way we discover we discover new drugs and new treatments, and we are basically convinced that the future of preclinical work is based on high throughput, and not only of high throughput of experimentation, but also high throughput of data. So I have a next slide here, which basically explains that the market is basically looking for, and everybody knows that, to speed up the clinical trial phases, to diminish the failure rates of clinical trials, and to minimize animal testing. And as I was saying, if you look at the grid on the right, we believe that the future is on the top right, which is basically a high throughput future with a high relevance, which means that complexity of organs on chips are going to be going up, but that high throughput needs to be the core focus, and that's, let's say, the core mission of NETRI is to democratize the organs on chips and leveraging existing equipments, and this will be a good example of that. We are a team of engineers, of biologists, of software developers, and user experience professionals, and we believe we need all 4 of those things to come together to make really impactful products. This is a bit of an overview of what you can find on our website, and you can see that we are right now in the bottom right, on the bottom left in the capabilities section, and we are going to be talking about functional activity, which in our case means electrophysiology. So what is that? For those of you who are not completely cognizant of those matters, basically neurons are the source and carriers of information. Okay, so their function is to produce electrical activity and to share it in a network. And very often, neurons are looked at by imagery. They're looked at in their geography, in the way that they are structured, as you can see on the right. But seldom do we actually look at the action potential and the way that the information is actually moved around. So there's a few people who do this really well. The main way to do this is by what we call microelectrode arrays, so electrophysiology using microelectrode arrays. So there's a few actors in this field, and mainly we have Axion, which is a company we are collaborating with, and you can see that microelectrode arrays are all those little electrodes that you can see in the middle bottom, and on the top right you can see those electrodes next to neurons. So the scale, you can see, like, the electrodes are about 100 to 50 microns wide, and neurons are about 10 microns wide, and you can see on the bottom the activity that you can see of those electrodes catching the signal and trying to make sense of that. So what electrophysiology and microelectrode arrays can do is basically assess the functionality of neurons, assess the excitability, the way that they can react to stimuli, whether they be electrical or they be from drug compounds, and the way that they connect with each other. And you can see there are different standard metrics that are being used in this field, the field of electrophysiology. Now, there's one large limitation that we have seen in this field, is that you often, it's as if you were looking at basically a petri dish on top of electrodes, and that will give you single population signals, or maybe a mix of populations inside something that is not fully representative of the complexity of a human body. And that, if it's great as a starting point, but we believe that in preclinical phases, we need to go one step further. And this is why we, as a company, develop organs on chip. So organs on chip. So what is that for basically applied to neural systems? Well, if you can see on the left, the neural system is actually distributed across the whole body. And so you can have different places where neurons live, and their axons, which is the long tail that actually distributes the signal, can actually live in your fingertips all the way up in your brain. And it's a distribution where the different places basically are different conditions, okay, and which you cannot recreate in an amalgam, in a mix of cells inside a petri dish. So this is where organs on chip come in, and you can see on the right there, our little white forms are the shapes of our chips. And the way they work is, you can see in green, you can see 3 channels. You have the left channel, the middle channel, and the right channel. And those are connected by microchannels, which are 3 micron wide channels that allow axons to sneak through and go to the other side and connect to the other population without allowing the body of the cells to actually go through. So they stay separate. And because those are really small, the microchannels are very small, you have fluidic isolation, which means you can culture a particular type of cells on one side with one medium, and a particular type of cells on the other side with another medium, and have them coexist and connect while they're not actually in the same medium. Okay, so that allows you to create neuron-to-neuron connectivity. So if you put neurons and neurons, but you could also put neurons and muscle, neurons and skin, neurons and anything, gut. There's all kinds of things that you can investigate by actually using this compartmentalization. Okay, so what you can see on the right is our standard chips, which actually come in this what we call quarter bento format, that go inside our 96 well plate format system that is basically standard and allows you to do all kinds of liquid handling and imaging and confocal imaging, and all the things that you might do with a normal 96 well plate you can do with our chips. So that's kind of the standard that we abide by, that makes our organs on chip quite desirable for preclinical phases. Now, the trick and why we're here is because what we talked about electrophysiology, we can mix that with our organs on chip. And this is where we use the power of Axion BioSystems technology to mix and to put a system of electrodes under our compartments, okay? And what that does is that it allows you to basically put, say, a type of cells on one side, a type of cells in the middle. This is a triolink, so this is the one where you can have 3 compartments. And here you can explore the whole gamut of the information going from your fingertips all the way to your spine and potentially all the way to your brain, okay? And the electrodes down there can record the activity and the information being passed from one place to another, but also you can disrupt the signal at any point in channel 1, in channel 2, in channel 3, okay? So this can be done like this. You can actually have other types of neurons there. You can also have only 2 chambers, for instance, and have, for instance, 2 parts of your brain. So glutamatergic neurons and dopaminergic neurons, or basically next to each other, not basically together on the same plate, which allows you to differentiate those 2 things and actually see the relationship between them. So what we have done with Axion is basically taken one of their sheets and they've adapted it to our layout, okay? And what you get is basically what you have on the right there, which is not a rendering, it's actually a photograph of the first we've made of a 16-chip with MEA recording, so 48 electrodes on each system. And these electrodes basically operate in 2 categories. There are the electrodes that you have that are in the channels that can see the intra-population network analysis, okay? And then there are electrodes that are between channels that actually sit on the microchannels. And those allow the inter-population network analysis, which basically will allow you to understand what happens to a population, but what is the effect of that population on the other. So if you have type A cells and type B cells, you can see the influence of A on B or B on A, depending if you disrupt A or disrupt B with either electrical signals or by disrupting them with a new compound or with a reference compound, and basically understand the mode of action or potentially the toxicity of one on the other by really understanding how things work, by differentiating them. We even have a chip that allows you to put synapses right in the middle, which allows you to only affect the synapses and not necessarily the cell bodies. So the type of output that this system yields is basically your standard electrophysiology output. So mean firing rate, burst rate, synchrony, the things that you know, but with the added advantage of knowing that those are compartmentalized, which means that you can see the synchrony of one versus the other, the burst rate of one versus the other. There's a lot more metrics that the fact that they're geographically confined adds to the system in terms of knowledge, okay? Then the other benefit is because we do have some, as you can see on the middle there, we have those red electrodes. Those are the interchannel electrodes, or the ones that are on the microchannels. Those I can actually see the information going back and forth from one side and the other. And what we posit here is that you'll be able to compute the directionality of the information and the speed of the information. Directionality can give you a lot of information on which way the information is flowing, and speed can give you the information, for instance, for myelinization or for the way that a compound affects the speed of travel of information across the system. Because they are completely isolated, and you can see here in the middle, you have the activity graphs, and you can see those really large spikes. That's because we have found out that on the microchannels, the signal to noise ratio is very, very high. So you get very, very distinct signals that you can monitor going from one side to the other and back. Kevin Gould, the CEO of Axion BioSystems, this is a testimonial for him, saying, modeling the complexities of the human brain and nervous system in vitro has historically been challenging, but organs-on-chip devices are transforming the study of neurological conditions and many other types of diseases. This collaboration with NETRI will provide scientists with innovative, high-throughput tools that accelerate scientists' breakthrough for researchers studying Alzheimer's disease, Parkinson's disease, epilepsy, and other disorders. So this echoes basically the way I've been thinking about this. Is you have on one side, you have people who have known organs on chip, who have not had access to electrophysiology. And on the other side, you have people who are quite familiar with electrophysiology, but who have never had the ability to compartmentalize, to further their research even more. And we believe that this association can create breakthroughs that are going to be quite astounding. And we're actually working on a little paper that we might be able to publish early next year. So this is basically the overview of the slide deck. I'll be happy to actually go back and forth and maybe discuss some of the different elements. Know that this is an early kind of release of what we're talking about. Right now we have beta versions that are available for purchase. The final system is actually due for Q1 2023. And yes, that's about it for this breakneck speed presentation. Next presentation will be about co-culture, and that'll be our number 5. I yield the floor to any questions that you may have in the chat section. Thank you.